So welcome back from the break. Let's take a look at the concept check question and then move on to talk about what we know about animal body plans. So summarize the four main stages of animal development. What family of control genes unique to animals play an important role in establishing animal body plans? Well, that's what we were just talking about in the last few slides. So the four main stages of animal development or embryological development are fertilization, not unique to animal development, but certainly the first stage of embryological development, followed by cleavage, where you have this modified form of cell division, where you have the division process taking place. You also have duplication of the DNA, but you don't have the intervening growth part of interphase. So basically you are subdividing the cytoplasmic volume of the egg into smaller and smaller compartments, the blastomeres that are the product of cleavage. Once cleavage is completed with the formation of the blastula, then you begin gastrulation with the complete reorganization of the embryo and the emergence of the germ tissue, which is going to give rise to all of the organ systems. And remember, the germ layers are ectoderm on the outside, mesoderm in the middle, endoderm on the inside lining the gut. Then following gastrulation, you have organogenesis with the emergence of organ systems, beginning with the neural tube and the notochord. The neural tube, remember, develops into the axial central nervous system. And I believe axial is the correct term there. Yeah, axial, because the nerve system that runs to the arms and legs is the appendicular system. So the axial nerve system, which is the central nervous system, including the brain, and then the notochord develops into the spinal column in vertebrates. So what are the control genes that are unique to animals? So remember, we talk about homeogenes that define body plans for organisms found in fungi, found in plants, found in animals. But the member of this family of control genes that is unique to animals are the Hox genes. When we know that there are 7 or 8 Hox genes, it depends on the nature of the cassette that you're dealing with. But in the simplest cassette, the foundational set, there are 7 genes. And we'll talk about the importance of those genes as we address the animal kingdom in more detail in the next 2 weeks with chapters 33 and 34. So let's move on. Okay, so we're not going to do body plans next. My mistake. I did go over this, and obviously I forgot that I'm incorporating phylogeny, which is the section that the chapter has got next. So let's talk about the phylogeny, then we'll talk about body plans, because body plans are kind of interesting, but let's talk a little bit about animal phylogeny. So based on molecular clock data, based on best guess and very, very limited fossil evidence, and I mean very limited fossil evidence in the form of microfossils that suggest that you're looking at primitive reproductive structures of animals. We believe that the common ancestor of the animal kingdom as we know it today probably emerged somewhere around 1 billion years ago. Now notice here on the slide that there is a window of time, nearly a 500 million year window for the possible time frame when the common ancestor of all animals first emerges. What we do know is that the best fossil evidence, or the most consistent fossil evidence for the presence of animals, doesn't really start to appear until about 560 million years ago, in a time period called the Ediacaran or Ediacaran. Now, this time period is named after a line of hills in the state of South Australia, which contains, or adjacent to this line of hills, is the original fossil bed for the Ediacaran. And there is some question, even in Australia, as to exactly how you should pronounce this name. Is it Ediacaran or Ediacaran? I've heard both. I prefer Ediacaran myself. That's my preference. And the reason why I say that is because I was very fortunate back in 2012 to actually meet the leader of the research group that is actively digging the Ediacaran fossil bed in South Australia. And I also had the opportunity to take a look at all the original fossils that they have taken out of this bed, to handle them, to examine them, and take as many photos as I wanted. I actually spent nearly a day in the South Australian Museum, where the research group is located, looking at these fossils, photographing them, and just spending some time with the fossils, and also talking with both the leader of the group and his assistant. I also got to talk with some of the other members of the group, because this group is not only digging in the Ediacaran fossil bed, they're also looking at a couple of Cambrian fossil beds as well. And I got a collection of trilobites from one of these researchers, just because they have they had all of these trilobites that they'd got out of the fossil bed. They didn't need them all because they had all the type fossils and other fossils that they needed for describing the organism. So they had more than they could use, and so they gave me a handful of these trilobite fossils. So that's where I got my trilobites from. These are very early Cambrian fossils. So that was kind of cool to be able to spend a day with these folk and just talk to them about what they were finding. Anyway, that's when we see the very first distinctive and definitive fossils that we can say these are probably animals. And again, I say probably animals because they don't look like any animals that you and I would recognize. And in fact, some people still argue that it's not really clear that these are actually animal fossils. But nonetheless, that is how the vast majority of the biological world interprets these. And you've seen some examples of them in the fossil lab, as you'll see when we get to the photos. But the protist ancestor that is likely to represent the origin of the animal kingdom is a group of colonial, I guess is the best term to use, protists known as choanoflagellates. These are probably the closest living relatives amongst the protists of all members of the animal kingdom, from the sponges onward. And the reason why can be seen in this diagram. Here is the colonial choanoflagellate. And so these individual cells are the individual organisms, but they live as a colony. And the cells have a main cell body with this tentacle-like arrangement known as a collar, with a protein net connecting these tentacles together, and then a single flagellum that emerges out of the middle of the collar. The activity of the flagellum creates a water current that draws water in and pumps it out through the net. And this allows the organism to capture food particles in the water, to phagocytose them, and through cytoplasmic streaming, move them into the body of the cell. Quite remarkably, there are a group of cells known as choanocytes or collar cells that line the open space inside a sponge called the spongiocele. And the activity of these cells creates the water currents that are drawn through a group of cells called porocytes that penetrate through the body of the sponge from the outside of the sponge into the spongiocele. And this column, or this water flow, allows the sponge to feed in much the same fashion. It sets up a flow of water through the net that links the tentacles together in the collar cell. and allows the sponge to filter particles out of the water, phagocytose them, and transfer the food particles via cytoplasmic streaming into the body of the choanocyte. Now, because a sponge is multicellular, as we'll see in chapter 33, we'll revisit this idea. Some of these food particles are then transferred to other cells that make up part of the body of the sponge, so that those food particles can be transferred to all of the cells that make up the sponge body. But it's the choanocyte and the essential identical form and function of the choanocyte compared to choanoflagellates that draws our attention to the choanoflagellates as the source of the common ancestor of the animal kingdom. That has been further reinforced through gene sequencing, because we've now found that some genes that are uniquely found in animals, outside of the Hox gene, some other genes that are known to be unique to animals, now have ancestral sequences in the choanoflagellates. Genes that are essentially identical or near identical to the same genes found in animals, indicating that the choanoflagellates share an evolutionary history at the genetic level, or an evolutionary connection at the genetic level with the animal kingdom. So they are considered to be a sister clade to the animal clade, and to diverge from the population of ancestral cells, that out of which would emerge the common ancestor of all animals, and would also emerge the ancestor of the choanoflagellates. So they're closely tied together. So if we look at the timeline for animals or the animal kingdom, remember again that we think that the emergence of the common ancestor to all animals lies back here, about 1 billion years ago. So we'll call this last universal common ancestor of all animals. Remember, Luca, we often refer to as the last universal common ancestor of all life. We talked about that back near the beginning of the semester. Well, we could also identify a Luca, last universal common ancestor of all animals. And that emerges somewhere around 1 billion years ago, maybe as far back as 1.2 billion years ago, maybe as recently as only 800 million years ago, but likely in the 1 billion year time range. And we have those microfossils of what could be animal embryos. That's the interpretation that's being given, coming out of fossil beds of this time range. But again, this is supposition. These are very, very small fossils and very hard to interpret, but we think, based on what they look like, that these may be early animal embryos. But our first clear emergence of animals is here in the Ediacaran. Then around 535 million years ago, we have the beginning of the Cambrian explosion. Remember, the Cambrian begins here at 542 million years ago. That's the boundary between the Ediacaran and the Cambrian. So the Cambrian is the time period that immediately follows the Ediacaran. We have this 10 million year period where we see the emergence of animal phyla, and certainly the diversification of bilaterals. That doesn't mean that bilaterals are not present in the Ediacaran. They most definitely are. So are radiata, animals with radial symmetry. But the diversification that occurs in the Cambrian explosion period is a diversification of bilaterals primarily. Then in the Silurian and following on from that, the Devonian, we see the emergence of vertebrates. first as developments from invertebrate chordates in the Silurian and perhaps even in the Ordovician. Predating the Silurian, we see the emergence of what are referred to as jawless vertebrates, conodonts, from the invertebrate clades that are part of the chordates. But in the Silurian and certainly in the Devonian, we see the emergence of fish as dominant vertebrates in the marine ecosystems, which are the only ecosystems that are present at this time. But around 340, 365 million years ago, we see the emergence of the first terrestrial tetrapods. But about 100 million years prior to that, around 460 million years ago, somewhere in that time frame, we actually see the first animals of any. So I'll put a bar in here. So approximately 460 MYA. Arthropods begin colonizing dry land. So if you ever wondered why insects are so successful and arthropods in terms of spiders, millipedes, centipedes, insects are so successful in a terrestrial environment, this is the reason why. Because while vertebrates started to colonize dry land around 360 million years ago, arthropods in the form of the early ancestors of spiders, centipedes, and millipedes started colonizing land 100 million years earlier, and basically had dry land to themselves to investigate all the available habitats and to evolve and adapt into all of those habitats, including the air, which is the reason why the vast majority of insects possess the ability to fly, whether they use it or not. Because again, they are the only flying animals for the best part of 100 million years. They're the only animals on dry land for 100 million years. And so once plants colonize land, and remember from the plant chapters, we know that plants started to colonize land approximately 470 million years ago, probably a little bit earlier than that, because we have the first, what we think are plant fossils around 470 million years ago. So insects are not really, or arthropods are not really going to start to colonize dry land until there's food resources in the form of plants. Then vertebrates come on shore almost certainly as a consequence of chasing down insect resources, arthropod resources, because amphibians are the first tetrapods to venture onto dry land and to start to utilize dry land. Of course, their big drawback is that they have to return to water to reproduce. But at the end of the Permian, which lies around here about 256 million years ago, actually probably a little bit closer in here, we have the end Permian. Extinction. And at the same time, we have all of the continental land masses as we know them today together in that single large continent, Pangaea. And so we have a period where we have not only an increase in the Earth's temperature, a major global warming event that raised the Earth's temperature at least 10 degrees Celsius and led to the loss of about 96% of all the existing species. But in losing all of those species, you also open up all the habitats that are available to organisms that can take advantage of them. And we know that there are reptile-like creatures here that are able to take advantage of these open niches. And so we see the emergence of reptiles, but we also see the emergence of the early ancestors of what would become the other major lineage of terrestrial tetrapods, the mammals. And so at the beginning of the Triassic, which is here at 251 million years ago, we see the emergence of reptiles, and ultimately towards the end of the Triassic, the emergence of dinosaurs, which would go on to rule the terrestrial habitats for the remainder of the Mesozoic until they become extinct in the end Cretaceous extinction about 66 to 65 million years ago, somewhere in that time frame. In the meantime, all through this time period, with the dinosaurs being present, mammals are also present. In the Jurassic and Cretaceous. They are small, they are nocturnal, they are insectivorous, they live in burrows, they spend their time mostly avoiding being eaten by the dinosaurs. But they're there. And in fact, we can probably thank the dinosaurs for our mammalian success ultimately, because it was the fact that mammals were forced to be nocturnal, were forced to live in burrows, were forced to chase insects at night as a food source that led to the development of the keen senses that mammals have, including the ability to high frequency sound, the ability to see well at night, probably even the ability to maintain an internal body temperature, something that we're not sure whether dinosaurs could do, but we certainly know reptiles can't do. However, we do know that birds maintain an internal high body temperature. So that gives us a suggestion that maybe some dinosaurs did. More of that to come in chapter 34, perhaps, if we have enough time. But we do know that mammals had to adapt to being nocturnal and insectivorous in order to avoid the dinosaurs. That would, at the end of the Cretaceous, with the death of the dinosaurs and the other accompanying large reptiles, the flying reptiles, the pterosaurs and pteranodons, the marine reptiles, the ichthyosaurs and the mosasaurs, basically the loss of all non-flying dinosaurs and dinosaur relatives. So the pteranodons, the tyrannosaurs, the mosasaurs, the ichthyosaurs. They're not dinosaurs, but they're reptiles. They all go extinct in the same mass extinction. The only dinosaurs to survive are the flying dinosaurs, which would eventually become the ancestors to birds as we know them today. In the Cenozoic, of course, with the loss of the dinosaurs, we have mammalian adaptation and diversification in response to that. It doesn't happen immediately. It takes about 10 million years for mammals to really start to diversify, but nonetheless, it begins with the loss of the dinosaurs and the loss of the other competing reptiles. And you see the success of mammals as a group as a consequence of this, including entering marine and freshwater habitats and becoming the dominant organisms and dominant predators, with perhaps the exception of competition against the sharks. But even there is evidence that vertebrates in the form of killer whales are competing with the sharks quite nicely. Thank you very much for dominance in a number of habitats, particularly around South Africa. We may get into that a little bit. when we get to that point. So basically, in diagramming this timeline, I've gone through the next set of slides, so I'm not going to spend a lot of time talking about them. I'll just stop at some pictures along the way. So here are some of the Ediacaran fauna. This one you should be familiar with because you will have seen this one in the fossil lab. I brought back a resin casting of the original fossil. So this is what we think is a fossilized jellyfish. That's the way it's been interpreted. We're not entirely sure what this is, other than it's bilateral and it appears to have a vertebral column. It's quite small, as you can see. So it's probably around 2 to 3 centimeters in length. And it's probably an early bilateral. It may not be a vertebrate, but it is because vertebrates don't emerge until much later in the scheme of things, but it is almost certainly a chordate because it's got this central support structure running down the center of it. So it's likely a chordate. That's likely a notochord. And we, of course, have other fossils of other types of what we think were animals. You've seen a couple of those again in the fossil lab. So that's the Ediacaran. Then we have the emergence of the Cambrian and the Cambrian explosion, that period of 10 million years where we see the diversification of the animal kingdom and the emergence of all the modern phyla that we recognize in the animal kingdom. So we think that the Cambrian is that point in time where one of two things are happening with respect to the phyla that we recognize today, of which there are about 35. And that is that either we see the emergence of those phyla during this time period, or that many of the phyla already had representatives present, but during this time period, we see an internal diversification within those phyla. It's not entirely clear from the fossil record which way it went, that the phyla were already present, but had a limited diversity, and the Cambrian explosion is simply an expansion of the diversity within all of the phyla, or whether this is a time period where many of the phyla actually diversified from their common ancestors during this time period. Irrespective of which way it goes, this is the time period where the modern animal kingdom, as we would recognize it, is established. But when we look at the fossils, those fossils don't look like any representatives of the phyla that we see today that we would recognize. These animals have very different forms, and it's taken a while to recognize that these actually do represent the modern phyla. And that's why there was a lot of thinking involved in trying to decide exactly what was the Cambrian explosion represents. Is it really an emergence of many new phyla that leads to a great diversification, or is it a diversification within the phyla that increases the presence of all the phyla? They're already there in a very limited sense, and this is just an increase. Again, we still don't really know, but we believe that that is the best interpretations that we can get. But what we do know is that at the end of the Cambrian explosion, all the phyla that are present today are present at that point, and that there are no phyla that emerge in the fossil record during this time period that are not present today. In other words, this is the period where we establish the modern animal kingdom in terms of the phyla diversity that we see today. So the other question that has yet to be fully answered is exactly what gave rise to the Cambrian explosion. And 3 hypotheses have been proposed to try and explain the origin of the explosion of diversity. So the first one is what is called an ecological hypothesis. And this is simply that there was a emergence of a predator-prey relationship. That if you take a look at the organisms that are present in the Ediacaran, a way of describing them is soft and squishy and easy to eat. And apparently, during the Ediacaran, there was very little evidence for predation, probably because the development of predatory devices would not take place until the Cambrian explosion. At least that's what the fossil record suggests. When we look at what is happening in the Cambrian explosion, we see an increase in organisms that clearly either have a predatory behavior or are trying to protect themselves from predatory behavior, because we see the appearance in the fossil record of hard parts. And hard parts are characteristic of either predatory behavior or protection against predatory behavior. And so as we see an increase in this predatory behavior and a predator-prey development of a predator-prey relationship, we also see a loss of the soft and squishy body plan that characterized the animals of the Ediacaran. So this is a change in the relationship between organisms, the emergence of predators, and the requirement for the prey then to respond to that predatory behavior by protecting themselves, an ecological hypothesis. And here is some evidence of that change in prey behavior. This borehole is characteristic of a type of mollusk that we have present today. This is a fossil that dates back to this time period in the Cambrian, but we see this type of neat round borehole as a predatory behavior amongst a group of mollusks that are extant today called moon snails. Moon snails are very active predators on other mollusks, and they create these neat round boreholes through the shells of other mollusks that allow them to access the soft body of the mollusk inside the shell. And of course, the mollusk inside the shell can't escape because it can't exit its shell, so it becomes a nice food source. But this type of predatory behavior apparently is a very old behavior, dating back at least 500 million years into the Cambrian. And specifically, we see it emerging in this period of the Cambrian explosion. Another cause, or another proposed cause that's been put forward, is the increase in oxygen leading to an increase in cell respiration, which of course leads to an increase in available energy. And that allows the organisms to become more active, to produce more body features, to create more complex body plans, and to invest resources in body structures that either allow them to be predators or allow them to protect themselves against predators. And we do know that there is an increase in atmospheric oxygen in this time period. So that would allow for a more diverse form of body plans, an increase in the size of body plans, an increase in the volume of body plans, and would allow for a greater activity. So this is our geological cause, an increase in oxygen. Then the third hypothesis is a genetic hypothesis that relates to an increase in the ability to utilize Hox genes and the increase in the importance of Hox genes in laying out body plans. And so the diversity of body plan, and it's just a simple increase in the variety of body plans, indicates that there is a genetic component here. And because the Hox genes are the genes that are involved in determining body plans, it almost certainly involved a change in the expression of the Hox genes, whether it be in the distribution of the Hox genes throughout the body of the embryo, or the timing of the expression of those Hox genes, the combinations of expressions of those Hox genes would be a driving force behind creating more diverse body plans. Now, when you think about these three hypotheses, they're not actually mutually exclusive. They actually are mutually supportive. If you're going to have an increase in the diversity of body plans and an increase in both predatory behavior, but also in the ability to protect yourself from predatory behavior, then you're going to need more energy to be a more aggressive predator, but you're also going to need more energy to create the body parts that are going to protect you from those predators. Those body parts are going to be a consequence of changes in gene expression. And as those body parts form part of the body plan, devices, spikes, shells, devices for crushing shells, and so forth. Those are all components of body plans, so they must be driven by changes in Hox gene expression. So the reality is none of these three hypotheses can really stand without an input from the other two. So it's most likely that all three of these causes were in play during that 10 million year period that we define as the Cambrian explosion. So this picture, which comes straight out of the textbook, and it's a very widely reproduced picture, is an artist's attempt to highlight some of the key species that are known from the Burgess Shale deposit, the original fossil bed in British Columbia in the foothills of the Canadian Rockies that represents the Cambrian time period, and specifically the early Cambrian explosion. So some of the creatures that you've got here, up here, you've got a chordate. So this is probably one of our very earliest ancestors, because we belong to the phylum Chordata, subphylum Vertebrata. So we are descended from the common ancestors of all, the common ancestor of all chordates. This is one of the very earliest chordates present in the fossil record. Then we have a little arthropod swimming over here, another arthropod swimming over here. I'll talk about this character in a moment. Got another arthropod down here, so you can see that there's a diversity of arthropods. This is called Hallucigenia. What's interesting about this one, and I think I might have mentioned this to you before, is that the original artist's conceptions of these, of the Hallucigenia, actually had them drawn upside down, because the paleontologists that originally described Hallucigenia got it wrong and thought that what were legs were actually protective spikes, and what were protective spikes were actually the critter's legs. And it took some fossils of Hallucigenia from another fossil bed that better preserved some of the fine details of the animal to finally allow paleontologists to realize that they'd got it wrong, and to invert the representation of Hallucigenia to put it on right side up again. Then you've got a bilateral burrowing worm. This is an important observation, because the only bilateral burrowing worms that are extant today are the annelid worms, and they're highly segmented, as you'll see when we talk about them in the next chapter. And that's an important observation, because when we think in terms of body plans, as you'll see, the segmented bilateral body plan with a true body cavity was always considered to be a more advanced form of body plan. But as you've seen, the bilateral body plan predates this. It goes all the way back into the Ediacaran, so it develops either shortly after the appearance of the animal kingdom in the form of the radiata, sponges, and then cnidarians and ctenophorans, or it parallels them. And so to have segmented worms with which is a very complex body plan, suggests that in fact body plans may not be a good measure of the phylogeny of the animal kingdom. A little bit more of that to come shortly. Then we have some sponges. We've got some early cnidarians here, and then we've got this guy. This is Anomalocaris, and it's the top predator of its day. When fossils of Anomalocaris were first found in the fossil bed, however, they were very broken up. And so the jaw structure was not attached to the body. These prey capturing devices were not attached to the body. And these were interpreted as actually being separate organisms. So what should have been a single organism was actually identified as parts of 3 different organisms. It wasn't until much later when more complete specimens of Anomalocaris came out of a new fossil bed representing a mid to late stage of the Cambrian, that it was recognized that these, what appeared to be parts of 3 different critters, were actually part of a single critter, Anomalocaris. And there were a number of different species of Anomalocaris. The smallest one was about the size of a large toenail. The largest one was, if I remember rightly, almost 1 meter in length. So there was a grand diversity of this group of animals, but they were very aggressive predators, and they are arthropods. So these are very early arthropods, and they were the top predators of their day. Then we see the emergence of early arthropods in the form of the ancestors of spiders, millipedes, and centipedes, transitioning onto dry land, beginning the colonization of dry land. Then we see the emergence of arthropods as a larger group, particularly in the form of insects. In the intervening time period, until around 360 million years ago, we see the emergence of vertebrates in the form of early amphibians. Then at the beginning of the Triassic, as I've already mentioned, beginning of the Mesozoic, at the beginning of the Triassic, we see a situation that favors the emergence of organisms that can tolerate high temperature and drier environments. And so we start to see early reptiles, out of which would emerge the dinosaurs. But and another branch of early terrestrial tetrapods in the early Triassic would come the cynodonts, which would evolve into the early ancestors of the mammals appearing in the fossil record late in the Triassic and early in the Jurassic as our little furball ancestors. Talked a little more to some degree about that in the previous diagram slide. Then with the death of the dinosaurs and about 70% of all other life forms, including a significant portion of the marine and freshwater life forms, we see the rise of the mammals. We see the rise of insects diversifying along with angiosperms. At the end of the Cretaceous and into the early Cenozoic, the diversification of insects and the diversification of flowering plants, as I mentioned back in chapter 30, seem to parallel each other. We're not entirely sure which came first. It's gone backwards and forwards, but the current thinking is that it was probably insects first and then flowering plants as a consequence. So let's take another break. And again, I'll make this a full break. So I'll meet you back on the other side of the break to answer this question. This is a pretty easy one. Put the following events in animal evolution in the correct order from oldest to most recent. Origin of mammals, evidence of the earliest evidence of terrestrial arthropods, emergence of the Ediacaran fauna, and the extinction of the large non-flying dinosaurs. See you on the other side of the break.